Hello students, welcome back to Gray's Gyan. Today we are diving into a topic that is absolutely fundamental to understanding how our body holds itself together at the microscopic level, and that topic is intercellular junctions. Now, think about this for a second. Your body has trillions of cells. What stops them from just falling apart? What keeps the lining of your gut sealed so that bacteria don't leak into your bloodstream? What allows your heart muscle cells to beat in perfect synchrony? The answer is intercellular junctions. So let's get into it. Intercellular junctions are specialized structures that connect cells to each other, and sometimes to the extracellular matrix beneath them. Now, what's really important to understand is that these junctions are not static, rigid structures. They are resilient. They can withstand incredible mechanical stress. Think about your skin being stretched, your gut constantly contracting, your heart beating 100,000 times a day. The cells stay together because of these junctions. Dynamic. They are constantly being assembled and disassembled in response to development, healing, and signaling. Absolutely essential. Without them, your epithelial tissues would simply dissociate into individual floating cells. No tissues. No organs. No life. The basolateral region of epithelial cell membranes is where most of this action happens, establishing junctions with neighboring cells and with structural components of the extracellular matrix. There are essentially 5 major types of intercellular junctions we need to understand: tight junctions, Zonula adherens, Desmosomes, Hemidesmosomes, and gap junctions. Let's go through each one. Before we get into individual junctions, let's talk about a very important concept: the apical junctional complex, or AJC. Look at this illustration from Gray's Anatomy. It beautifully shows how junctions are arranged from the top to the bottom of an epithelial cell. At the very top, the apical region, you have 2 circumferential belts running parallel to each other like 2 rings around the cell. First, the tight junction, also called Zonula occludens. Second, just below it, the Zonula adherens. Both of these belts are associated with F-actin, a cytoskeletal protein. Together, they do 2 crucial things. Number 1, they control epithelial permeability, deciding what can pass between cells. Number 2, they determine epithelial cell polarity. And this is a key concept. The apical cell domain, the top of the cell, sits above these belts. The basolateral cell domain, the sides and bottom, sits below the belts. This boundary is not arbitrary. The apical surface faces the lumen, or the exterior. The basolateral surface faces the basement membrane and neighboring cells. Each domain has completely different proteins, channels, and functions. This polarity is what allows, for example, your intestinal cells to absorb nutrients on one side and release them into the bloodstream on the other. Now let's zoom into the tight junction, also called the Zonula occludens or occluding junction. The name tells you exactly what it does: it occludes, it seals. Imagine 2 neighboring epithelial cells. At their apical border, proteins from one cell literally stitch into proteins of the adjacent cell, creating a watertight seal between them. This prevents molecules from sneaking through the space between cells, what we call the paracellular route. The key proteins here are claudins, the primary sealing proteins. Different epithelia express different claudins depending on how tight the barrier needs to be. Occludin works alongside claudins. JAMs, junctional adhesion molecules. And on the cytoplasmic side, scaffolding proteins ZO1, ZO2, and ZO3, which anchor the whole complex to F-actin. Now here's why this matters clinically. In inflammatory bowel disease, conditions like Crohn's and ulcerative colitis, tight junction proteins like claudins are disrupted. This allows luminal bacteria and antigens to leak across the gut wall, triggering chronic inflammation. This is sometimes called leaky gut at the cellular level. Pathogens are also smart. Many bacteria and viruses specifically target tight junction proteins to gain entry into the body. So the tight junction is literally your body's first line of cellular defense. Just below the tight junction sits the second belt of the apical junctional complex, the Zonula adherens, or Adherens junction belt. If the tight junction is about sealing, the Zonula adherens is about holding and organizing. The key transmembrane protein here is Cadherin, and this is a calcium-dependent protein. That's important. Remove calcium from the system, and these junctions fall apart. That's why EDTA, which chelates calcium, is used in the lab to detach cell cultures. Cadherin on one cell binds Cadherin on the neighboring cell in a homophilic interaction, like a Trax-like. Their cytoplasmic tails connect to beta-catenin and alpha-catenin, which then link to F-actin, the contractile cytoskeleton. This creates a circumferential contractile belt around the entire cell, and when these belts contract across an epithelial sheet, it can cause the sheet to fold and bend, which is critical during embryonic development, for example in neural tube formation. The other important complex here is the aphidin-nectin complex, which helps establish the junction before cadherins consolidate it. Now remember the polarity concept. This belt, together with the tight junction above it, creates the boundary that separates the apical domain from the basolateral domain. This is the structural basis of epithelial cell polarity. Alright, now we move to a junction that is an absolute exam favorite, the desmosome, also known by its beautiful Latin name, macula adherens, meaning spot of adhesion. And that name is perfect. Because unlike the tight junction and Zonula adherens which form continuous belts, desmosomes are spot-like. They look like rivets or spot welds scattered across the lateral surfaces of cells. Here's the analogy I want you to remember. Desmosomes are like the rivets on a pair of jeans. They provide incredibly strong, localized points of attachment. Now, the molecular structure is beautiful. On the cell surface, you have desmoglein and desmocolin. These are cadherin family proteins. They reach across from one cell and grab onto the corresponding proteins of the neighboring cell. On the inside of each cell, they connect to a dense cytoplasmic plaque made of desmoplakin, plakoglobin, and plakophilin. And critically, this plaque connects to intermediate filaments, specifically keratin in epithelial cells and desmin in cardiac muscle. This is a huge distinction to remember. Desmosomes link to intermediate filaments, not actin. Write this down. It comes up in every exam. This creates a transcellular network of intermediate filaments, a true mechanical scaffold running through the entire epithelial sheet. And this is why desmosomes are most abundant in tissues that face the greatest mechanical stress. The skin and the heart. Clinically, when desmosomes are attacked, the consequences are dramatic. Pemphigus vulgaris, autoantibodies against desmoglein 3 destroy desmosomes in the skin and mucous membranes, causing painful, widespread blistering. Nikolsky's sign is positive. Arrhythmogenic cardiomyopathy. Mutations in desmoplakin or plakophilin cause cardiac desmosomes to fail, leading to fibrofatty replacement of the myocardium and life-threatening arrhythmias. Now, what if I told you there's a structure that looks like half a desmosome? That's exactly what a hemidesmosome is. While desmosomes connect cell to cell, hemidesmosomes connect cells to the basal lamina, the specialized extracellular matrix that underlies all epithelial sheets. Think of hemidesmosomes as the anchor bolts that attach the floor of your epithelium to the foundation beneath it. The key difference from desmosomes is in the transmembrane protein. Instead of cadherins, hemidesmosomes use integrin alpha-6 beta-4, which binds to laminin in the basal lamina. They also involve BPAG1 and BPAG2, bullous pemphigoid antigens, and like desmosomes, they connect to intermediate filaments on the intracellular side. Clinically, this is where two important blistering diseases come from. Bullous pemphigoid, the most common autoimmune blistering disease. Autoantibodies attack BPAG2, also called BP180 or BPAG1, destroying hemidesmosomes, causing subepidermal blisters. Tense blisters on normal or red skin. Nikolsky's sign is negative, unlike pemphigus. Epidermolysis bullosa, genetic mutations in hemidesmosome proteins, skin blisters with the slightest touch. A devastating condition in children. So remember the key distinction. Pemphigus equals desmosome attacked equals intraepidermal blister. Pemphigoid equals hemidesmosome attacked equals subepidermal blister. And now, my personal favorite, the gap junction. Gap junctions are completely unique. They are the only junction that provides direct cytoplasmic communication between adjacent cells. And here's what makes them special: they are not linked to the cytoskeleton at all. So how do they work? Let me walk you through it step by step. Step 1. 6 proteins called connexons assemble in the plasma membrane of one cell, arranging themselves in a ring to form a hollow cylinder. This cylinder is called a connexon, or hemichannel. Step 2. The neighboring cell does the same thing, assembles its own connexon. Step 3. The 2 connexons from adjacent cells dock head-to-head across the narrow gap between cells. Step 4. Together, they form a complete aqueous channel, directly linking the cytoplasm of both cells. Now, what can pass through this channel? Small molecules, ions like calcium and potassium, amino acids, sugars, second messengers like CAMP, essentially anything under about 1 kilodalton in size. The most spectacular example of gap junction function is in the heart. Cardiac muscle cells are connected by gap junctions rich in connexin 43. The electrical signal for contraction spreads instantly through these channels, which is why the entire heart contracts as one coordinated unit. If you lose connexin 43, you get arrhythmias. Gap junctions are also critical in embryonic development, allowing signaling molecules to coordinate the fate of developing cells, and in neural signaling, where they mediate electrical synapses. The key phrase to remember: gap junctions equals communication junctions equals connexins equals not cytoskeletal. Now let's take a step back and look at the molecular players, the cell adhesion molecules, or CAMs, that make all these junctions possible. There are 4 major families. Number 1, Cadherins. These are the calcium-dependent adhesion molecules. E-cadherin in epithelial cells, N-cadherin in neurons, V-cadherin in blood vessels. They bind homophilically, E-cadherin binds E-cadherin. On the inside, they connect to actin via the catenine family. Loss of E-cadherin is a hallmark of cancer invasion. It triggers what we call epithelial to mesenchymal transition, or EMT. Number 2, Integrins. These connect cells to the extracellular matrix, collagen, laminin, fibronectin. They are heterodimers, made of an alpha and a beta subunit, with over 24 different combinations, each with a specific binding partner. They signal bidirectionally, from inside the cell out, and from outside the cell in. Number 3, IgCAMs, the immunoglobulin superfamily. These are calcium-independent adhesion molecules. They include NCAM in the nervous system, ICAM and VCAM in the vasculature, and JAMs at tight junctions. Critically important in neural development and immune cell trafficking. Number 4, Selectins. These are lectin-like proteins that bind carbohydrates. L-selectin on lymphocytes, P-selectin on platelets, E-selectin on endothelium. They mediate the rolling of white blood cells along the vessel wall, the first step in inflammation. They are major drug targets for anti-inflammatory therapy. Now let's go back to this beautiful illustration from Gray's Anatomy, Figure 1.28, and appreciate what it's showing us. On the left, you have the schematic diagram, showing the full hierarchy of junctions from the apical domain at the top to the basal lamina at the bottom. You can see the tight junction at the apex, followed by the Zonula adherens with its actin, then the macula adherens, the desmosome, with its intermediate filaments, and at the base, the hemidesmosome anchoring to the basal lamina. On the right, you have the actual electron micrographs, real images from real tissue. Panel A, a transmission electron micrograph showing the apical junctional complex. You can see the electron-dense regions where the tight junction and adherens junction form. Panel B, a freeze fracture preparation of a tight junction, showing the characteristic network of sealing strands on the membrane face. Panel C, a transmission electron micrograph of a hemidesmosome. You can see how the epithelial cell base attaches to the basal lamina below. Panel D, a freeze fracture image of a gap junction, showing the beautiful cluster of connexon particles packed together like a plaque. When you look at histology slides in your exams, knowing what to look for at the electron microscope level is what separates a good answer from an excellent one. Let's now talk about what happens when junctions fail, and why this matters enormously for clinical medicine. Inflammatory bowel disease. Tight junction disruption allows luminal contents to breach the gut barrier. Claudin proteins are downregulated. The result: chronic, uncontrolled inflammation. Pemphigus vulgaris. IgG antibodies destroy desmoglein 3. Desmosomes break apart. Keratinocytes lose their connections. The epidermis separates within itself, causing painful intraepidermal blisters and erosions. Without immunosuppression, this disease was once fatal. Bullous pemphigoid. Antibodies attack BPAG2 at the hemidesmosome. The entire epidermis separates from the dermis. Tense fluid-filled blisters on normal skin. More common in the elderly. Cardiac arrhythmias. Loss of connexon 43 at gap junctions breaks the electrical coupling between cardiomyocytes. The organized spread of the action potential fails. Potentially life-threatening dysrhythmias result. Cancer invasion. Loss of cadherin at Adherens junctions triggers EMT, epithelial to mesenchymal transition. Cancer cells lose their adhesion, gain motility, and invade surrounding tissues, called metastasis. Cadherin is literally called a tumor suppressor. Arrhythmogenic cardiomyopathy. Mutations in desmoplakin and plakophilin cause cardiac desmosome failure. Fibrofatty infiltration of the myocardium, a major cause of sudden cardiac death in young athletes. Understanding junctions is not just basic science, it is directly clinically applicable. Let's consolidate everything we've learned into one clean comparison. Tight junctions, apical belt, proteins, claudin, occludin, zeoproteins, links to F-actin, function, seal and define polarity. Zonula adherens, just below, also a belt, proteins, cadherin, cadherins, links to F-actin, function, adhesion and mechanical integrity. Desmosomes, basolateral spots, proteins, desmoglein, desmocolin, desmoplakin, links to intermediate filaments, function, resist mechanical stress. Hemidesmosomes, basal surface only, proteins, integrin alpha-6 beta-4 PAGs, links to intermediate filaments, function, anchor cells to basal lamina. Gap junctions throughout, proteins, connexons forming connexons, not linked to cytoskeleton, function, direct intercellular communication. Print this table. Stick it on your wall. These 5 rows will save you in any histology or pathology exam. Before we wrap up, here are your 6 golden takeaways. 1. Tight junctions and zonula adherens together form the apical junctional complex, the master organizer of epithelial polarity. 2. Desmosomes are spot welds linked to intermediate filaments, not actin. This comes up constantly. 3. Hemidesmosomes are half desmosomes that use integrins to anchor cells to the basal lamina. 4. Gap junctions are the only communicating junctions, using connexons, and they are not connected to the cytoskeleton. 5. Know your CAMs, Cadherins are calcium dependent. Integrins bind ECM. IgCAMs are calcium independent. Selectins mediate rolling adhesion. 6. The disease associations, pemphigus, desmoglein, intraepidermal blister. Pemphigoid, hemidesmosome, subepidermal blister. Cadherin loss, cancer invasion. And that brings us to the end of today's lecture on intercellular junctions. I hope this session has helped you understand not just the what, but the why behind these structures. How they're built, how they function, and what happens when they break down. Anatomy is not just memorization, it's understanding the logic of the human body. And once you see that logic, it never leaves you. If you found this helpful, please like this video. It genuinely helps the channel reach more medical students like you. Subscribe to Gray's Gyan so you never miss a lecture.